Click the bell icon and subscribe to our channel so that you will not miss any classes.
Hey everyone, let us continue to study the microbiomolecules which are present in acid soluble pool, that is lipids and nucleotides. First let us study the lipids. Lipids are generally water insoluble. That means when we put in water, they will not be solubilized, they will stay separate. And the lipids could be of three different types. They could be simple fatty acids, or they could be glycerol, or they could be cholesterol. In your textbook, they mentioned or they give an explanation about simple fatty acids and glycerol, and they given the structure of only cholesterol. So now I am giving little explanation about cholesterol and detailed explanation about simple fatty acids and glycerol. And I suggest you to keep NCERT textbook with you so that you can have a comparison and meaning of each and every word within that. First let us study the simple fatty acids. The fatty acids are represented by a general formula COOH, that is carboxyl group attached with the R group. That explanation is given in your text. So that can be represented as carboxyl group attached with the R group. And here R group is variable to form different kinds of fatty acids. If the R group is methyl, if it could be methyl group, that is CH3, or R group could be ethyl, C2H5, or R group could be the higher number of CH2 groups, and higher number means you may ask question how many numbers of CH2 group may present. The number ranges between 1 to 19 carbons. So they have mentioned two examples here. For example, in palmitic acid, the number of CH2 group is 14, and the formula is written as CH3 CH2 14 carboxyl group. Here this is the R group, and this R group contains 14 CH2 group and 1 CH3. So total there is presence of 16 carbon including the carboxyl carbon. This point is very important. 16 carbons are present along with carboxyl carbon. And another example they have mentioned of arachidonic acid. Arachidonic acid have 20 carbons including the carboxyl carbon. Next, let us study the classification of classification of fatty acids. Fatty acids are classified as saturated and unsaturated fatty acids. Saturated fatty acids, those do not have a carbon to carbon double bond, and unsaturated are those which contain one or more carbon to carbon double bonds in them. They are called unsaturated fatty acids. And another type of the lipids, as we discussed, are glycerol. Glycerols are referred to as trihydroxypropane. Glycerol are called trihydroxypropane, and the formula is written as CH3OH, CH2OH, CH3OH. So here trihydroxy refers to there is presence of 3 OH groups. And many lipids, for that matter, they contain both fatty acid and glycerol. So fatty acid, in some there may be presence of only fatty acid, in some there may be presence of only glycerol. But many of the lipids, many a times which are occurring in living systems, will have both fatty acids and glycerol in them. So how fatty acid and glycerol are linked with one another, that is very, very important. And your NCERT text, they have given one statement, that is always the fatty acids are found esterified with glycerol, which means that always the fatty acids is linked with glycerol through a ester bond. That is the meaning of esterified. So the number of fatty acids could be linked with the glycerol will ranges from 1 to 3, because there is presence of 3 replaceable OH groups here. 3 OH groups could be substituted by 1 fatty acid or 2 fatty acids or all the 3 fatty acids together. So, depending upon the number of fatty acids linked with the glycerol, fatty acids are classified, the lipids are classified as monoglycerides. Monoglyceride means here CH3, OH, CH2OH, CH3OH, that is trihydroxypropane. Instead of one OH, there is presence of one fatty acid linked to it. So that is called monoglyceride. And you can understand now on your own that diglyceride means there is presence of 2 fatty acids. Triglycerides, there is presence of 3 fatty acids. So monoglyceride, diglyceride and triglyceride. And remember, always this name is given depending upon the number of fatty acids present with the glycerol, which means that in the lipids which contain both fatty acids and lipid, there is always presence of one glycerol. So this point you need to understand, it's very important. And the number of fatty acids may be 1, 2 or 3, not more than 3, because there are only 3 OH group. So that is about the diglycerides, triglycerides and monoglyceride we studied. And in your diagrammatic part, they have given the structure of a triglyceride as this. And it is a triglyceride. Here you could see this is R1, that is fatty acid linked with the R group, carboxyl group linked with the R group, which means fatty acid. This is fatty acid 1, this is fatty acid 2 and this is fatty acid 3. So it is a triglyceride. Now, this triglyceride could be a conjugate lipids. Conjugate lipid means apart from the pure lipid part, there may be presence of some inorganic groups in them. And one example is given, that is of phospholipids. Phospholipids are those which have the lipids which are containing phosphorus or phosphorylated organic compounds. Lipids which contain phosphorus or phosphorylated organic compounds in them. And example for a phospholipid is lecithin. And one structure of the phospholipid is given in text, and I'm going to write that CH2OC R1 and here CHO C double bond R2, CH3O C phosphate double bond OH, O CH2 CH3 and here N CH3 CH3 and here is one more CH3. So here is a phospholipid because it is containing a phosphate group in it. So that's about the explanation of fatty acids and lipids given. And one more thing you should know, the difference between oils and fats. Oils and fats are nothing but types of lipids where the oils will remain as liquid or oily form at winter, that is at low temperature, and example is gingerly oil. And fats are those they will become solid at winter, and example is butter. So this is one thing which is given in text, so you need to remember this also. And another type of a lipid that they mentioned, but they have not given explanation, they have just given the structure of cholesterol. Cholesterol are the types of lipids which contain a 27 carbon containing compound, which is attached to the hydroxyl group, that is hydroxyl means OH group. So this is called a cholesterol, and cholesterol are the essential components of animal cells and animal cell membranes. So usually They are found in animal cell membranes. And what you need to remember about the cholesterol according to NCERT textbook is the structure. This is very important, so I'll write this. And this is the structure of cholesterol which is given in NCERT textbook, so you could refer the page number 107. And you have to remember all the structures of the compounds. Microbiomolecules is given in NCERT textbook, page number 107. So you have to remember all of these, very important. And that's all about lipids. Now let us study the another interesting microbiomolecule found in the living system is nucleotides. Nucleotides are also a carbon compounds, which means what you meant by carbon compounds, that is they are also biomolecules, because in initial classes we have discussed that all carbon containing compounds are called biomolecules. So this is the meaning. Carbon compounds means they are biomolecules and they contain heterocyclic ring in them. This is very important. They contain heterocyclic rings in their structure, and these heterocyclic ring containing structure could be called a nitrogenous bases. Nitrogenous bases alone will not form a nucleotide. So to form a nucleotide, it requires three components. They are pentose sugar, nitrogenous base, and a phosphate group. Pentose sugar is different in DNA and RNA. So that means one more point you have to concentrate here. Nucleotides are the monomers of nucleic acids. Nucleic acids means DNA and RNA. So you have studied previously that amino acids are the monomeric units of proteins. Simple sugars are the monomeric units of carbohydrates. So likewise, nucleotides are the monomeric units of nucleic acids, and nucleic acids are of two types, DNA and RNA. And you have to remember here, if the nucleotide is found in DNA, then it contains a pentose sugar called deoxyribose sugar. Here I am referring pentose sugar means it contains 5 carbon. So let me write the structure here. This is the pentose sugar and it contains CH, OH, and here H, OH, and here CH2, OH, and here is H. So here the number of carbons are important. So you have to remember here is the first carbon, second, third, fourth, and out the ring 5th carbon. Here it is called deoxyribose sugar because at second carbon there is lacking of one oxygen. That's why deoxyribose sugar. Same way, the sugar which is present in RNA is ribose sugar. And ribose sugar is also a pentose sugar. So let me write the structure here. Pentose sugar, here H, OH, here H, OH, H, OH, and here out the ring CH2OH, and here H. So the number of carbons, first, second, third, fourth, and fifth. Here at third carbon and second carbon, or majorly at second carbon, there is no oxygen lacking. Oxygen is present. That's why it is called ribose sugar. And to this ribose sugar, there is a attachment of nitrogenous base at a first carbon. At first carbon, through the formation of N-glycosidic linkage, which means in DNA here there is an attachment of nitrogenous base. This is first carbon, and here there is attachment of nitrogenous base through the formation of, here there is a formation of N-glycosidic linkage while forming a nucleotide. And the nitrogenous bases present in DNA and RNA are found to be of two different types, and they are purines and pyrimidines. Purines are the nitrogenous bases which contain two heterocyclic ring in them. So two heterocyclic ring containing pure nitrogenous bases are called purines. So the structure goes like this. There is presence of two heterocyclic ring. And pyrimidines have only one heterocyclic ring in them. And the purines are of two types. They are adenine and guanine. And pyrimidines are of three types: thymine, cytosine, and uracil. So here, according to NCERT textbook, so I need to go. Adenine and uracil structure is given. Let me write that later, little. Here, one more point I need to discuss is, in case of DNA and RNA, there is presence of only 4 type of nitrogenous bases. So here I have written 5 nitrogenous bases, right? Then what are the 4 types of nitrogenous bases present in DNA? They are adenine, guanine, thymine, cytosine in DNA, whereas in RNA, adenine, guanine, uracil and cytosine. That means in DNA, instead of uracil, thymine is present. That is the meaning. So what are all the structure you need to remember is of adenine and uracil. And the structure of adenine is like this. So let me write the structure in different color. So here is the structure of purine. Purines contain 2 heterocyclic ring, as I mentioned earlier. Here is nitrogen containing NH2. Here N double bond. And here N, NH. And here is double bond and one more N here. So this structure you have to remember. So no need to go through all the structures. Here what are the structures mentioned in the NCERT text, so that you need to remember. This is structure of adenine. And structure of uracil is also given. Uracil is a pyrimidine which contain 1 heterocyclic ring. And the structure goes like this: NH, double bond O, NH. And this is the structure of uracil. And now the third important component of a nucleotide is phosphate group. And phosphate group is always derived from the phosphoric acid, that is H3PO4. H3PO4 get linked with a pentose sugar by the formation of a ester bond. So I'll write later the complete structure of one nucleotide. And here is the structure of phosphoric acid. So H3PO4, P double bond O, OH, OH, okay, H3PO4, here one more OH. Okay, so that is the structure of phosphoric acid. Now let us understand how nucleotides are exactly being formed. Nucleotides are formed when there is a attachment of phosphate group and phosphate group and nitrogenous base to the pentose sugar. First, there is a attachment of nitrogenous base. So here is nitrogenous base to the pentose sugar through the formation of N-glycosidic linkage. And this structure is called nucleoside. You should recall that there are 5 different types of nitrogenous bases present, right? So there is a possibility of formation of 5 nucleosides, and the name goes like this. They are adenosine, that is when adenine get attached with the pentose sugar. Same way, guanosine, thymidine, uridine, and cytidine. And these are the nucleosides. Now, when to form a nucleotide, to this nucleoside, if there is an attachment of phosphate group at the 5th carbon, always phosphate group is attached to the 5th carbon by the formation of a ester bond. Here ester bond is formed, and now this is called a nucleotide. And again, phosphate group is only of one type, but nitrogenous bases attached to it will be of 3 different types. Hence, there is a possibility of formation of 5 different types of nucleotides. And the name of nucleotides include adenylic acid, thymidyllic acid, and remember all these names are given in NCERT, so you should remember guanylyc acid, uridylic acid, and cytidyllic acid. And you may ask question, how to remember the name, which one is a nucleoside and nucleotide. One thing you should remember, if the names are ending with syn, the name is of purine. Purine names will end with syn, and pyrimidine's name will end with din. So you have to remember adenine and guanine are purines, so they form adenosine, guanosine, like that. So thymidine, cytidine, uridine, if it is end with, then it is called nucleoside. So here one more trick to remember the names of nucleotides is phosphate group is derived from phosphoric acid, right? So phosphoric acid, if it get linked with a pentose sugar, then it will form an acid. So if acid comes, then it is nucleotide. If there is no acid, then it is nucleoside. So that is one way of remembering the name. And here one nucleotide structure is given in your textbook, that is of adenylic acid. So here structure go like this. Here is adenine, and here is phosphate group linked with it, OCH2. Here phosphate, double bond O, OH, and here is OH, and here is ester bond, here is N-glycosidic bond. You have to observe in textbook that here at both second and third carbon there is presence of oxygen, which means that this nucleotide is present in RNA. So this is a ribose sugar. If ribose sugar is present, then that nucleotide is found in RNA. That is understood. So if they remove this, then that nucleotide will be found in DNA. That is the trick to remember. So that's all given in NCERT textbook about nucleotides. In next class, let us continue with primary and secondary metabolites.